Hello dear friends, welcome in this video lecture. Friends, we have started to learn to study about the bioinorganic chemistry. As we know that the bioinorganic chemistry is the interdisciplinary branch of biology and chemistry, and in particular inorganic chemistry. That is, it involves the study or the interactions of metals or different elements found in the nature in the different life processes. Okay, in previous lecture, in last lecture, we have discussed about the different role of different elements in the biological processes, how they are controlling the life processes, what are their functions in main or important physiological processes, okay, and some part about metalloproteins. Today our topic of discussion is metalloenzymes. So these metalloenzymes are nothing but these are the special class of metalloproteins. Sorry. So the metalloproteins which act as a catalyst in specific biological reactions, in specific biochemical reactions, we can say these metalloproteins are known as metalloenzymes. Okay, so as we know that enzymes are the biological catalyst, or they act as a catalyst in different life reactions, different biological processes, biological reactions, in such a way that in the similar way the metalloenzymes are also acting as a catalyst in different biochemical reactions. But the speciality is that these enzymes are containing metal ions. These enzymes are containing metal ions or metal atoms. Okay, such are called as metalloenzymes. Now there are number of metalloenzymes can be found in different life processes. Okay, today we will discuss about some of these. Okay, the first one is hydrolytic enzymes. The first type of metalloenzymes are known as hydrolytic enzymes. Again, these hydrolytic enzymes are of again performing different kinds of biological reactions. Hydrolytic enzymes mainly involved in the either addition or removal of an element from the water molecule which is involved in the biochemical reactions. So what is the main important function of these hydrolytic enzymes? So hydrolysis, we know the meaning of hydrolysis, the reaction with water. Okay, so on the similar way, the meaning of hydrolytic enzymes are, or what is the function of these hydrolytic enzymes? This is nothing but they are involving in the addition or removal of elements of water molecule. So what are the different elements of water molecule? Only two, that is either hydrogen and oxygen. When addition or removal of hydrogen or oxygen from the substrate molecule is done, okay, that reactions are known as hydrolytic reactions, and hydrolysis reactions involve hydrolytic enzymes, and these hydrolytic enzymes are metalloenzymes. Okay, so we will see one by one how the different chemical reactions involve hydrolysis. First one is the enzyme known as carboxypeptidase. The enzyme known as carboxypeptidase, it is also functioning as a hydrolytic enzyme, and the reaction is it removes terminal amino acids from the protein molecule. Okay, we know that proteins are polypeptides. They are having number of amino acids bind together by peptide bond or amide bond, and the enzymes known as carboxypeptidase, they are involving in the hydrolysis of these proteins. Okay, so when NHCO bond is broken, okay, this NH This moiety is containing one amino acid and this moiety is containing another amino acid. So these two amino acids are joined together by a amide bond, or we call it as a peptide bond. So when the breaking of this NH-CO bond will takes place, the one protein, one amino acid will get broken, will get separate from that polypeptide chain or protein. And for this, there is a the function of enzyme, or that enzyme which is functioning this type of reactions is known as carboxypeptidase. You have to remember these things. You have to remember the names of the enzymes that are participating in such specific type of biochemical reactions. Okay, because in your examination the MCQ type of questions may be asked, and such specific answers or such specific questions may be asked, which type of enzymes, metalloenzymes specifically, are involving in the removal of terminal amino acids from the protein chain. Okay, next one is carbonic anhydrase. Carbonic, we know that formula of carbonic acid is H2CO3, which is formed by the combination of water and CO2 molecule in the nature or in the water. Okay, so when dissolved carbon dioxide present in the water body, when it is react with water molecule, it is forming carbonic acid H2CO3. Okay, so this is the formation of carbonic acid from water and CO2, and again dissociation of that carbonic acid into its constituents, that is H plus and carbonate ions, bicarbonate ions. Okay, so this reaction is catalyzed by the carbonic anhydrase metalloenzyme. Carbonic anhydrase metalloenzyme. Okay, next one is dehydrogenases. So what is the function of such enzymes? The function is known as dehydrogenases, means removal of hydrogen. D means removal, hydrogen means hydrogen atom. Okay, so when there are some chemical reactions which involve the removal of hydrogen atoms, such as this one, the enzymes helping in such biochemical reactions are known as liver alcohol dehydrogenase. The name of the enzyme is liver alcohol dehydrogenase. So this is the alcohol, that is ethyl alcohol, present in the liver. When NAD plus reacts with this, it removes hydrogen atom from this ethyl alcohol, and it is get converted to acetaldehyde molecule. Okay, and the consequent byproducts are NADH and H plus. Okay, next one is called metal nucleotide chemistry. In metal nucleotide chemistry, the type of enzyme involved is ribonucleotide reductase. Ribonucleotide reductase means nucleotide is get reduced, okay, with the help of this enzyme, and the example is very famous, that is ribose nucleic acid is get converted to deoxyribose. Ribose is get converted to deoxyribose. Okay, means what? Oxygen is get removed. Ribose get converted to deoxyribose means oxygen is get removed, and removal of oxygen is known as reduction. Okay, so that's why the name is ribonucleotide reductase. The next type of metalloenzymes is known as glucose isomerase. Glucose isomerase means the function is known as isomerization. So we know that isomers having same chemical formula but different structural formulae. Okay, these are known as isomers, and D-glucose and D-fructose are the isomers of each other. They are only having same molecular formula, but they are differing only one functional group, that is D-glucose is aldohexose and D-fructose is aldoketose. Okay, so that's why the isomerization of this D-glucose into D-fructose is achieved with the help of glucose isomerase enzyme, and the title this function is known as isomerization process. So this is one kind of metalloenzymes known as hydrolytic enzymes. Okay, now the next type of metalloenzymes that are taking part in different biochemical reactions are two-electron redox enzymes. So we know that redox reactions are nothing but oxidation reduction reactions, and in these redox reactions, oxidation state of a particular element is generally decrease or increase by 2 units. When increase or decrease of oxidation state is done, or it is happening, or it is occurring by 2 units, then such reactions are known as 2-electron redox reactions, and the enzymes which are helping in such reactions are known as 2-electron redox enzymes. Okay. For example, the very famous example is Metalloenzyme known as nitrate reductase. So this is the enzyme. The name of the enzyme is given as it is involving in particular type of biochemical reactions. So as the name is including nitrate, means it is reducing nitrate, that is NO3 minus into NO2 minus. So NO3 minus is get reduced to NO2 minus. Okay, so this is known as nitrate reductase enzyme. Now the next type of metalloenzyme, third type of metalloenzyme is known as multi-electron pair redox enzymes. Multi-electron pair means more than one electron pair. One electron pair means 2 electrons, that we have seen in the previous type. Okay, 2-electron redox enzymes. But here some of the biochemical reactions are such that they are involving redox reactions which involve more than one pair of electron. Okay. For example, here the 2 examples are given for 2-electron pair. So 2 electron pairs means total 4 electrons are involved in such redox reactions. For example, when dioxygen is reacting with 4 H plus ions, there is a combination of 4 electrons, and these 4 electrons are nothing but 2 electron pairs. Okay, and there is a formation of 2 water molecules. In the similar way, backward reaction can also be possible with the same enzyme, metalloenzyme. Okay, then this catechol type of moiety is there, having carboxylic group at one carbon of the benzene ring. Okay, when oxygen reacts with this such type of moiety, such type of functional groups, then rupture or breaking of this carbon-carbon double bond in the aromatic ring is done with the help of 2-electron pair metalloenzyme. Okay, such reactions are not possible chemically in the laboratory. Okay, but such reactions can be possible only in biochemistry or in biological processes, and this is done with the help of metalloenzymes. Okay, and this is get converted to such carboxylic acid derivative. So it is a tricarboxylic acid derivative which is obtained from the catechol derivative. Now there are some 3-electron pair biochemical reactions, 3-electron pair redox reactions, 3-electron pair redox reactions. So what is the example? Atmospheric nitrogen when reacts with 6 protons in presence of 6 electrons. So 6 electrons are totally it is nothing but 3 electron pairs. Okay, so there is a formation of 2 ammonia molecules, and this is one of the very famous reaction that we can say that it is a part of nitrogen fixation, N2 fixation. Okay, so nitrogen is present in the atmosphere and it is get fixed. Okay, and it is 3-electron pair redox reaction into ammonia. Another example involving 3-electron pair redox reaction is nitrite ion combines with 4 protons and 6 electrons, formation of NH4 plus, that is ammonium ion formation takes place with the help of with the removal of 2 water molecules. So all these are multi-electron pair redox enzymes. These are biochemical reactions which are controlled by the metalloenzymes. Now the last type of metalloenzymes involving some chemical reactions known as rearrangement reactions. So we have already studied in organic chemistry, different rearrangement reactions are possible where there is no any net charge developed in the substrate or in the product. Such reactions are known as rearrangement reactions, and the rearrangement reactions involving metalloenzymes are also found in the biological processes. For example, the 1,2 carbon shift that is possible with the help of such enzymes that require vitamin B12, So in absence of that vitamin B12, that enzymes are unable to work, that enzymes are unable to function their role in that 1,2 rearrangement, 1,2 carbon shift or 1,2 methyl shift, let us say. Okay, such rearrangement reactions are possible only and only when there is a presence of vitamin B12 in that biological process. We know that vitamin B12 is an complex molecule which consists of a corine ring, just like porphyrin ring. There is a presence of 4 cyclic corine ring, and in that corine ring, the central part of the corine ring is occupied by the cobalt metal. And this cobalt metal is get bonded to the carbon atom, and hence this carbon atom means there is a cyanide moiety is there, and we can say that COCN bond is present in the vitamin B12 structure. Okay, and such metal carbon bond is present, and hence we can say that vitamin B12 is a organometallic compound. Okay, so what you have to remember here, vitamin B12 structure consists of a corine ring, okay, which is made up of cyclic nitrogen containing functions, and the central portion of that vitamin B12 is consisting of cobalt metal, okay, and it is bonded to cyanide, that is CN moiety, CN functional groups, okay, and hence the name of vitamin B12 is also called as cyanocobalamin. Okay, this is the reason why vitamin B12 is called as cyanocobalamin. Okay, in your MCQ type of examination, such questions may be asked like which central metal ion or atom is present in the vitamin B12 structure. So the answer is cobalt. Which bond is present? Mainly why it is called as organometallic? Because the cobalt metal ion is get bonded to the cyanide group in the vitamin B12 structure, hence it is known as organometallic compound. Okay, so this is the summary of metalloenzymes that we have studied in the bioinorganic chemistry. I hope you enjoyed this video. Thanks for watching. Have a nice day.